before and after, do you sense that you accomplish your goal of finding more freedom here, like of the long-haired professor? Were you able to come here and have a symposium with a bunch of academics and discuss
Yeah.
freely all topics?
Oh yeah, I had. I always enjoyed discussing academically, but once it became time to become a professor, um, I lost my will to do so because I felt niched in. You know, I was working on zebrafish angiogenesis, brain angiogenesis, which is very
Wow.
important. Zebrafish is just a model organism, but many of the brain development parts are very similar to how human brains develop, and understanding how the blood vessels are growing around the brain and into the brain to support the brain with nutrients and oxygens later on. That's a very important field. Understanding that is very important. But I still felt I was too much niched in. So I thought, in science, there's like 4 or 5 other people that understand what I'm doing, and I'm not reaching a larger audience. So I went out of science, and
All
and
right,
then I
sorry,
went
one
to
second.
yeah,
Sorry to interrupt. A lot of information here. So
yeah.
you are a professor. Are you a professor?
I'm not a professor. I could have been a professor. At the point when I would apply for professorship, I left academia.
Okay.
Yeah,
So you were also doing research.
yeah.
Are you still doing research or you were doing it before?
No, I was doing it before. I'm not doing
Okay.
research anymore.
So you were researching neurogenesis, which is creating, basically rebuilding the brain, essentially, like the brain creating more new connections, new neurons.
Yeah, for example, I was focusing on the blood vessels that if you want to understand, that people, you know, for example, if people have a brain tumor, it's important to understand how the blood vessels grow because they support the brain tumor with nutrients. If you want to get rid of the brain tumor, you need to understand ways to get rid of the blood vessels. For example, people get aneurysms in brain where blood vessel just breaks or bursts, and then you need to understand, maybe some blood vessels, maybe they have weaker walls than other blood vessels. And to understand the blood vessels, you need to understand how they develop. I think that's the most straightforward ways to understand something. You want to see how it's created. And the advantage of zebrafish is they are transparent, so you can basically and the blood vessels support the brain after 2 days. So you basically get a large part of zebrafish embryos. You just watch them develop with a microscope, and after 1 or 2 days, you can watch how the blood vessels grow into the brain. That's what I did.
Zebrafish.
Yeah,
Zebrafish.
yeah,
Okay, I
yeah.
see.
So
Wow. Okay.
yeah,
Wow. Okay. What did you learn? What was the conclusion of that experiment?
we learned that there was a gene called RECK, which is a gene that is upregulated in many cancer, especially cancers that are progressing into later stages. And this gene
Upregulated.
actually,
Sorry.
upregulated. Okay, so the gene is switched on, or let's say in general, it shows abnormal activity in a lot of the cancers, and so we thought that this gene regulates. This gene is responsible for regulating the blood vessels, because if it has an abnormal activity, there's like a crazy overgrowth of blood vessels. That's basically what we found, and we found that it's not just like some crazy overgrowth everywhere, but that if we have a mutation, we had a zebrafish that didn't have the functioning REC gene. And we compared that zebrafish without that gene to a control zebrafish, and then you can see, okay, the differences between the blood vessels in both fishes, in both versions, that is probably what that gene is responsible for. And we found that without this RECK gene, certain blood vessels weren't developing at all. And so I characterized that, and we found certain the role that REC plays in development of these blood vessels is probably switching on some signal in the cell that makes these cells grow.
Okay.
So that's what we found. Yeah. Yeah,
Wow. Okay.
and the interesting thing about that was that it was very, very specific, right? It was a very specific subset of blood vessels in the brain not the entirely blood. The rest of the fish was fine. In the front of the brain, blood vessels were fine. Everywhere else in the body, blood vessels were fine, but just in the back of the brain, those blood vessels weren't there. And so that gave us interesting new insight. Also, if you think about it, that means that there's a curious sharing of work between some genes are responsible for blood vessel development in the back of the brain, some genes are responsible in the front of the brain, in the mid of the brain, and it's really interesting to characterize all that. And that's what I basically started to develop. But for some reason, I couldn't see myself being a professor at that and really pushing it forward because I felt I was too much in a niche, and then I would really do my thing without anybody else really knowing anything about it. And so I stepped out of academia. But does that make sense? That was what we were working on.
Yes, yes, definitely. That's wild. So a gene malfunction is basically connected to like a certain growth of blood vessels in a certain part of the brain, basically.
Yeah, yeah,
Wow.
yeah.
Okay. All right. What? Okay. So